Hello students, welcome back to my channel. Today I am going to discuss the multiple choice question for the NEET aspiration animal cell, body architecture at the cell stage, that is the animal cell. The cell theory of Sheldon and Shawn stated that cells are the fundamental structural units of plants and animals. All cells are living, all cells have nuclei, cells reproduce by mitosis and meiosis. The correct option, the first one, the cells are fundamental structural units of plants and animals. Next one, the first person to see a cell under the microscope was Matthew Sheldon, Robert Hooke, Antonin Leeuwenhoek, Theodor Schwann. The correct option is Robert Hooke. Next, cell membrane in animals is composed mainly of the molecules of lipids, and proteins, carbohydrate. Correct option is lipids and proteins. Which of the following is the best explanation of earlier discovery of nucleus than the cell theory? Options: nucleus is a more important structure than cytoplasm. Robert Brown had a better microscope. Nuclei take a darker stain than other parts of a cell. Nucleus is a visible body while cell theory is an abstract generalization. Fourth option, last one, nucleus is a visible body while cell theory is an abstract generalization. A plant cell differs from an animal cell in the absence of endoplasmic reticulum, mitochondria, centrioles, ribosome. Correct option is centrioles. Ribosomes are centers for protein synthesis, photosynthesis, fat synthesis, respiration. Excuse me. Correct option is protein synthesis. The cell theory postulated by Sheldon and Shawn is not correct, neither new or wrong, new but wrong, not new but incomplete, yet the credit should be given to the Prospers for being able to attract the attention of biologists of their times. Fourth option is right. Which statement reflects the correct position of cell theory? Cells have independent lives within higher forms of life. New cells arise by a process resembling crystal formation. Prospers of cell theory were not able to attract the attention of the biologists of their time. Cell theory was proposed by Sheldon and Shawn in 1838-39. Last option is correct. Cell theory was proposed by Sheldon and Shawn in 1838-39. Cell organelles which have electron transport system are rich in catabolic enzymes and are regarded as powerhouses of cells are mitochondria, nucleus, centrioles, endoplasmic reticulum. Correct option is mitochondria. In electron microscope, the source of light used is ordinary daylight, ultraviolet, infrared beam of electron. The correct option is beam of electron. DNA is most concentrated in nucleus, nucleolus, chromatin, plastid. It is in chromatin. Mitochondria are associated with the function of cellular digestion, excretion, protein synthesis, cellular respiration. It is associated with cellular respiration. Plasma membrane is impermeable, formed of cellulose, selective barrier, non-selective barrier. Plasma membrane is a selective barrier. The main difference between animal and plant cell is that first, a plant cell lack rigid cell wall, animal cells lack rigid cell wall, animal cells possess large vacuole, plant cells possess small vacuole. Correct? Animal cells lack rigid cell wall. Lysosomes are reservoirs of fats, RNA, hydrolytic enzymes, secretory of glycoprotein. Correct option, hydrolytic enzymes. Electron microscope has revealed the presence of ribosomes, chloroplasts, chromosomes, chromoplasts. Correct option is ribosomes. Correct option is ribosomes. Next one, lysosomes are responsible for protein synthesis, digestion of organic molecules, oxidation of organic molecules, transmission of hereditary characters. The correct option is digestion of organic molecules. Cellular organelles containing hydrolytic enzymes are ribosomes, microsomes, lysosomes, mesosomes. Correct option is lysosome. Cell theory was propounded by Morgan, Haldane, Robert Hooke, Sheldon, and Shawn. Correct option is Sheldon and Shawn. Endoplasmic reticulum is main center of excretion, carbohydrate synthesis, protein synthesis, blood formation. Correct option is protein synthesis. Animal cell is limited outside by basement membrane, cell wall, plasma membrane, shell membrane. It is plasma membrane. Function of centrosome is inhibition of cell division, initiation of cell division, to provide site of cell division. None of it. It is initiation of cell division. The word cell was coined by Robert Hooke, Weizmann, Cuvier, Charles Darwin. Correct option is Robert Hooke. Fluid filled in cells are extracellular, intracellular, interstitial, tissue fluid. Correct option is intracellular. DNA is present in lysosomes, Golgi complex, chromosomes, endoplasmic reticulum. Correct option is chromosomes. Endoplasmic reticulum present in nucleus, chromosomes, DNA, cytoplasm. It is present in cytoplasm. Centrosome is a nuclear structure of animal cells, cytoplasmic structure of animal cells, cytoplasmic structure of both plant and animal cells, cytoplasmic structure of plant cell. Correct option is cytoplasmic structure of animal cell. Cellular respiration, that is transformation of chemical energy into usable bioenergy, takes place in lysosomes, ribosomes, mitochondria, endoplasmic reticulum. Correct option is mitochondria. Wall of mitochondrion is made up of lipids, lipoproteins, proteins, carbohydrate. Correct option is lipoproteins. Mitochondria are the seat of cellular respiration, ATP formation, electron transport, all of these. All of these. Mitochondria are concerned with the ATP formation, ADP formation, glucose formation, none of this. Correct option is ATP formation. Genes are made up of histones, hydrocarbons, polynucleotides, lipoproteins. Correct option is polynucleotides. Cell membrane is made up of glycoproteins, phospholipids and proteins, phosphoproteins, double layer of protein. Correct option is phospholipids and protein. Mitochondria in a cell are concerned with the formation of ATP and from ADP, ADP from pyruvic acid, lactic acid from citric acid, ADP from ATP. Its correct option is ATP from ADP. Protein synthesis occur upon Golgi complex, ribosomes, lysosomes, mitochondria. Correct option is ribosomes. Next one, mitochondria will be found in abundance in the cells of the tissues having maximum activity, wound healing activity, minimum activity, average activity. Correct option is maximum activity. Which of these is not a part of a nucleus? Nucleoplasm, chromatin, karyosome, centrosome. Centrosome is not a part of nucleus. Mitochondria are not found in maturing RBCs, mature RBCs, nerve cells, liver cell, that is in mature RBCs. DNA is found in nucleus only, cytoplasm only, both, none of these. The correct option, DNA is found in both. It is found in both. Extranuclear extranuclear DNA is found in ribosomes, mitochondria, lysosomes, Golgi. It is mitochondria. Nucleus is separated from surrounding cytoplasm by a nuclear envelope, which is single and non-porous, double and non-porous, single and porous, double and porous. It is double and porous. Suicidal bags of cells are ribosomes, Golgi bodies, lysosomes, nucleoli, contractile vacuole. Correct option is lysosome. Cell organelles associated with the secretion are Golgi complex, ribosomes, chloroplast, mitochondria. Correct option is Golgi complex. Intake of fluid by a cell is known as phagocytosis, pinocytosis, exocytosis, none of these. It is known as pinocytosis. Nucleus was discovered by Robert Brown, Robert Hooke, Dujardin, Purkinje. Nucleus is discovered by Robert Brown. Cristae are cell organelles, ground subsurface of mitochondria, folds of inner walls of mitochondria, outer membrane of mitochondria. It is the folds of inner wall of mitochondria. Tubulin protein occurs in digestive enzymes, microtubules, rough endoplasmic reticulum, thylakoid. It is in microtubules. A polysome is a group of lysosomes, mitochondria, cell, ribosome. It is a group of ribosome. Ribosomes can be observed in a cell by a light microscope, electron microscope, both of these none. It is by the electron microscope. To determine the ultrastructure of a cell organelle, the most likely method of successful observation would be phase contrast microscopy, autoradiography, light microscopy, electron microscopy. It is electron microscopy. Ribosomes were discovered by Palade, Porter, Didieu, Colliker. It is discovered by Palade. An organelle shows a highly folded inner wall. Ultrasonic disruption of an organelle yields fragments which can synthesize ATP. The organelle is chloroplast, ribosomes, mitochondria, and correct option is mitochondria. Cell wall is impermeable, permeable, semi-permeable. Cell wall is cell wall is permeable, impermeable. Ribosomes consist of DNA and proteins, RNA and proteins, RNA and amino acids, RNA and DNA. It is consist of RNA and proteins. The term The term chromosome was introduced by Sanger, Vanden, Waldeyer, Altman. It is introduced by Waldeyer. Electron microscope differs from ordinary light microscope in having high resolving power due to the use of fast electrons having a shorter wavelength than that of ordinary light rays as the source of illumination, gamma rays as the source of illumination, ultraviolet rays as the source of illumination, electrons having longer wavelength than ordinary light as the source of illumination. Correct answer is the first option, fast electrons having a shorter wavelength than that of ordinary light rays as the source of illumination. 58. An angstrom, the unit of measurement in transmission of electron microscopy, is equal to 0.01 millimicron, 0.0001 millimicron, 0.0001 millimicron, 0.001. Correct option is 3, 0.0001 millimicron. A delimiting outer membrane is absent in plastids, nucleolus, mitochondria, lysosome, that is in nucleolus. 1 millimicron is equal to 10 to the minus 8 millimeter, 10 to the minus 7 millimeter, 10 to the minus 5 millimeter, 10 to the power minus 6. It is correct option, 10 to the power minus 6 millimeter. Golgi complex are derived from endoplasmic reticulum, cytoplasm, nuclear membrane, cell membrane. It is endoplasmic reticulum. Hydrolytic enzymes are found in mitochondria, ribosomes, peroxisomes, lysosome. Correct option is lysosome. Electron microscope is better than a light microscope because it has high energy electrons, greater focal length, more magnifying power. Light energy is not used. Correct option is more magnifying power. Primary function of rough endoplasmic reticulum is synthesis of lipids, protein, starch, cell wall. It is protein. The fluid mosaic model of the structure of the plasma membrane was given by Robertson, Daniel and Evson, Singer and Nicholson. None of these. Correct option, Singer and Nicholson. Catalase enzyme is found in peroxisomes, chloroplast, mitochondria, cytoplasm. Correct option is peroxisome. How many angstroms make a meter? 10 to the power 6, 10 to the power 10, 10 to the power 3, 10 to the power 9. The correct option is 10 to the power 10. Which of these must be used for a detailed study of live cells? Ultra microscope, TEM, SEM, phase contrast microscope. Correct option, phase contrast microscope. Smallest cells are far known are bacteria, PPLOS, blue-green algae, human egg. It is PPLOS. Fluid mosaic model of the structure of plasma membrane was postulated by Robertson, Daniel and Evson, Overton, Singer and Nicholson. It is Singer and Nicholson. Glyocalyx is found upon inner surface of plasmalemma and is found in of proteins, found upon outer surface of plasmalemma and is formed of carbohydrate, found upon outer surface of plasmalemma and is formed of glycolipids and glycoproteins, found upon outer surface of plasmalemma and is formed of phospholipids and carbohydrate. The correct option is found upon outer surface of plasmalemma and is formed of glycolipids and glycoprotein. Cell drinking Cell drinking precisely involves the process of phagocytosis, pinocytosis, exocytosis, and cell drinking, it is pinocytosis, right? The fluid phase of cytoplasm is called cytosol, hyaloplasm, cytoplasmic matrix, all of this. It is all of this. Microtrabecular lattice is found in connective tissue and forms a supporting framework, bones and renders long bone spongy cells and form a cytoskeleton, lungs and forms the lung array. It is cells and forms a cytoskeleton. A channel system in cell architecture is provided by Golgi complex, microtubules, endoplasmic reticulum, microfilament. It is endoplasmic reticulum. Metabolic enzymes which bring about the synthesis of chemical components of unit membrane in cells are found in mitochondria, lysosomes, SER, RER, smooth endoplasmic reticulum, rough endoplasmic. It is found in smooth endoplasmic reticulum. Dictyosomes in cells are units of smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi complex, food reserve. They are in Golgi complex. Lysosome in a cell are formed by endoplasmic reticulum, mitochondria, dictyosomes, none of this. It is the dictyosomes. In mitochondria, Christie acts as sites for oxidation reduction reaction, breakdown of macromolecules, phosphorylation of flavoproteins, protein synthesis. It is oxidation reduction reaction. Which organelle is located near nucleus and contains a collection of flattened membrane-bound cisternae? Mitochondrion, centriole, Golgi apparatus, nucleus. Correct option is Golgi apparatus. Inner membrane of mitochondrion is usually high convoluted, forming a series of unfoldings known as grana, cresti, thylakoids, lamellae. Correct option is cresti. Besides giving our secretory vesicles, Golgi apparatus is also concerned with the formation of cell plate after cell division in plant, plastids, lysosomes, grana of chloride. The correct option is lysosome. LY, lysosomes. Lysosomes were discovered by D. Dew, Robert Brown, Hook, Robinson. They by the D. Dew. I hope everything is clear. Any doubt anywhere, you can ask me in the comment section. Again, we will meet in the next video. Thank you.